I want to extend this notion of information into the proteins. So proteins, you remember, are the polymer of amino acids. And they have quite a lot of similarities in their notion of information coding than the nucleic acids do. They have a kind of a backbone, and then they have information that is associated with the backbone. When we write proteins, we designate N or NH2 or amino as one end of the protein molecule. And then we write amino acid 1 joined to amino acid 2, amino acid 3, and then we have C or COOH or carboxy, and that would be at the other end. Okay? The amino acids are joined together by a peptide bond, covalent bonds between the amino group and the carboxyl group of the amino acids. And this NC bond will form a kind of a backbone, similar to the nucleic acids. Not the same at all, but the notion is similar, as I'll show you in a moment. The amino acid order is relative to the two ends of the molecule. So there is polarity to proteins that is based on the amino and carboxy ends, and is also based on the amino acid order. And we can draw a parallel, again, to nucleic acid structure. The carboxy amino acid is the last added, and the next one will add onto the carboxy end. So that the carboxy end has the last amino acid added, and the next adds to this carboxy end. A little compressed down there, but let's go to the slide. The first thing I want to show you is that proteins have got two different ends, an amino end and a carboxy end, with the amino acids arrayed between those. And then if we look at the protein polymer, two different amino acids here, glycine and alanine. I've circled the alpha carbon on these, join together to make a peptide bond, which I've circled in blue, and leaving a free amino end on the first amino acid of the polymer and a free carboxyl end on the last amino acid. And if a new one were to add, this is just a dipeptide, a very tiny protein. If the next amino acid were to add, it would add where I've arrowed the incoming next amino acid. When we write proteins, we write with the 3 or the 1-letter amino acid code. We always, and this has another parallel to nucleic acids, we always write the amino end and the carboxy end. You never get too old to write the amino acid and the carboxy end onto a protein. So here I've got a couple of example peptides, little tiny proteins with their amino and carboxy ends indicated. And here again, the direction of polymerization. The order of the amino acids is the information. The polarity of the amino acid chain, the N and the C ends, shows the first to the last amino acid added, and the direction to read the information. If we compare the proteins and the nucleic acids, there is an order of monomers that can be read in a defined direction, and that defined direction is the information. In the nucleic acids, it's the order of the nucleotides from 5' to 3'. In the proteins, it's the order of amino acids from the free amino to the free carboxy terminus.